Hello, everybody. My name is Matthew Hershey. Thank you for coming. I am excited to join you today with Akshay Bareja to tell you a little bit about a gene expression analysis. Continuing a little bit from 2 months ago, we're going to show you an end-to-end analysis of gene expression. So it's going to be pretty nitty-gritty into the data science for molecular biology. But for those of you who are interested in this field and really want to understand gene expression analysis, this is a great place to start. Before we get going today, I just want to tell you one thing that we're quite excited about. The first is that we are expanding the scope of our tidy biology efforts. And one of the things that that means is that we're going to be changing the name a little bit, and we're going to expand the types of content that we cover. So we've previously focused exclusively on molecular biology and biochemistry, but more recently we've been expanding some of these efforts to include some clinical data sets, data science for physicians, and really up and down the spectrum of different sciences. So we're calling this bespoke data science, or bespokeDS. One of the major and most exciting things that Akshay and I are working on is to try to identify the ways that we can enhance learning experiences in data science. And so one of the things that we've been able to do is generate bespoke or tailored custom data sets for data science. So gone are the days, we think, of trying to learn data science with test data sets. And we will usher in this new age of learning data science using your own data. So we're quite excited about this, so stay tuned for some of those announcements. But that's for another day. So today we have Akshay Bareja. Akshay, welcome. So tell us a little bit about what you're going to be describing today.
Hi, Matt. So today we're going to go through how to perform a differential expression analysis using a given microarray dataset that we'll be downloading from the Gene Expression Omnibus. And as you mentioned, this will be end-to-end, so from downloading it to getting a list of significantly differentially expressed genes.
Excellent. All right. Well, let's go ahead and dive right in. Just as a note, I will go ahead and man the Q&A board while Akshay is going through some of these demos. So if you guys have any questions, go ahead and pipe in. So Akshay, it's all yours.
Thanks, Matt. All right. Welcome, everyone. So again, before we begin doing the analysis, just make sure that your Teddy Biology Meetup project is up to date. So open up your project, go to your Git pane, and click on the blue poll button just to make sure everything's up to date. Mine already is, so I'll go ahead and begin the analysis. So this is slightly different from what we've been doing previously. Previously, we've been using R Markdown files, but here we'll just be using a simple R script. So if you go into the folder 2020_07, there's just one file in there, and that's the R script you need to open. Before going through the actual code, let's. So I should say the expression, the dataset we're analyzing has this ID GSE 29297. So before we actually go through the code, let's just Google that. And if you Google it, it'll take you, the top link will take you to the relevant page on the Geo site, the Gene Expression Omnibus. So it's the top hit. And if you click on that, this gives you you'll see a summary of the experiment. So in this case, it's a Nature Immunology paper from 2011, in which the researchers, what they did was they wanted to identify a gene that establishes quiescence in T cells. And in this case, they homed in on TSC1, tuberous sclerosis complex 1. And they showed that establishing quiescence is essential for maintaining survival of these cells. So so as we all know, TSC1 is a tumor suppressor, and it forms the link between AKT and the mTOR complex 1. And so they found that when you deleted TSC1, you had an overactivation of mTORC1, and that resulted in an increase in apoptosis. So if you scroll down that page, you see this button that says Analyze with GO2R. If you click on it, you can see these are all the various samples that were included in this microarray dataset. And GO2R, if you've not used it before, is a really easy way of performing a differential expression analysis. So what you do is, so in this case, we have all of these samples. So the first thing we do is we define groups. So in our example script today, what we're doing is comparing wild type activated cells with these TSC1 deficient activated cells. So the activated cells were cells that were given a stimulus to activate the T cell receptor. So to do that, you first need to click on the define groups. And in this case, since I've done this previously, it remembers what I've done. I made two groups, control and test. Now, to make a group is easy. Just call it something, and you press enter, and it creates a new group. So what you then do is you click on the necessary samples, and you assign them to a given group. So in this case, I'm assigning these three samples to my control group, and these three to my test group. Great, so now I have 3 control and 3 test, and I can do a simple comparison of these two groups. Now, if you scroll further down and click on R script, GO2R automatically updates an R script that compares these two groups. And so all I did was I cheated and I copy pasted all of this code into R, and we'll be going through this line by line. So if you go back to your project, let's go ahead. And so we need 3 packages to do this analysis: BioBase and GeoQuery to download the dataset and process it. And we'll be using this Lemma package to perform the actual differential expression analysis. So, the shortcut is Control Enter to run anything in your R script in your console. Line 8, so we use this getGeo function to download the relevant dataset, so this GSC 29297. I'm going to skip that because I've already done it. And you see that it saves it as a dataset called gset. Now, this is of type, it's a large list. And if we click on it, we can see, click on this play button, you see there are a lot of, there's different types of information in this rich gset dataset. Line 9, basically that, so in most instances, you will have, for each dataset, you'll have one platform. So platform is basically the chip type. So in this case, it was an Affymetrix chip type. I forget which one specifically. But in case, for whatever reason, the researchers decided to use multiple platforms using the same biological samples, that would be included in the data set. So this line searches for that. So if there's more than one, you have to specify the name of the specific platform you want to use. But if it's just one, there's only one platform to use. So in this case, it ran through the if-else statement, and it called IDX. It gave it a value of 1. So there's only one platform here. Line 10 subsets gset using 1. So it's gset brackets 1. And what that does is, so if we go ahead and run it, you'll see that it's converted gset from a large list to a large expression set. And we click on the large expression set, it looks very similar to the previous version of gset. It's just that it's changed the type of the data set. So it's changed it to an S4 type. Now, whether or not you've known this, you have previously used base type. So like, for example, base type. And you've used S3 objects also, like factors in S3 object. S4 is a more complicated object type in R that's used a lot by various bioconductor packages. And it's a really useful, standardized way of including a lot of different information in one object type. It's a little difficult to navigate or get used to, but once you get used to it, you see that there's a nice logic to it. So that's what line 10 does. And I should say that after copy pasting all the code, I added a lot of comments, and these are really for my benefit, but hopefully you'll find them useful also. If you skip to line 20, that's again just a line that is provided by Geo. And what it does is it imports a lot of the column names that are within your GSET dataset, or it makes those available for use later on. Uh, it'll include these names, these column names, into our final data output table, which is called the top table. So we'll go ahead and just run that. And actually, I'll copy paste this make names function, just execute it in the console, and you can see, so these are the column names I was referring to. So if you don't include this step, it's fine. You'll just get a list of genes with p-values and fold changes, but you won't have all this other useful information like the gene ID and so on. So the first thing we want to do is, if you remember, when I selected the control and treatment groups in GO2R, I just clicked on them. So line 23 is kind of the coding equivalent of clicking. So you see that it has all of these samples, and all the ones marked X are the ones we don't want. The ones marked 0 are the control group, and the ones marked 1 are the treatment group. So it's difficult to type this out, but if you did what I did and just clicked on the relevant groups, GO2R automatically will give you this. So we go ahead and run this. And what we then want to do is we want to take this string and we want to split it into individual X's and zeros and ones. And to do that, the first thing we do is we'll make an empty list called SML. And then we'll write this for statement that goes through this GSMS string and one by one takes out the X's, zeros, and ones. And so if you look at SML, you see there are 16 constituents. What we then want to do is we want to select all the ones that aren't an X, because the X's mark the samples you don't want to include in your dataset. So we'll use this which statement here, or this which function, and we'll save it as an object called cell. And you see cell contains the indices of the samples we want to select. And so subset SML with this cell. And then finally, we subset the GSET object with cell. So we see, so now this GSET object will only contain the samples that we want to use for our analysis. Now, line 33, you can use this. We use this expression function to get basically the expression values for the samples of interest, and we call that X. And if I go ahead and just run head X in the console, you see that it's a simple rectangular object. So we have 6 columns because we chose 6 samples, and the rows refer to the probes that are on this Affymetrix chip. Line 34 is I really like this function. It's a way of determining whether or not your data have already been log transformed. Because to do the Lemma analysis, which is the differential expression analysis, it's expecting log transform data. So in line 34, the first thing you do is you work out these various quantiles, what they are, and you save it as an object called QX. And if we run QX in the console, you see that these are the respective, so the 99th quantile refers to the value 11.27 and so on. And so this statement here from line 35 to 37, what it's doing is it's actually not, it's kind of using a yardstick. It's searching for values that are of a certain threshold. So what it's saying, for example, here, QX5, which is the 99th quantile, it's asking if it's above 100. Because if it's above 100, as I've said here, it probably has already been log transformed. Because if it, let's see, let me get this right. So it's seeing if the value really is greater than 100. Because if it is, then the anti-log value will be 2 to the power of 100, which is a incredibly large number. It's highly unlikely that that's the case. So if any of these statements end up being true, then your data probably haven't been log transformed. You should log transform it. So we go ahead and run that. And if we just type out log C, sorry. in the console, we see that it's false. So we don't need to run. These values have not been log transformed. So if we go ahead and run this if. Sorry, we have to run all of it. This if statement, it'll log transform the values. I also copy pasted these lines here, lines 45 to 48, and that's just the base version of plotting a box plot. And that just shows us the distributions in each sample, and it shows that they're quite comparable, and so we can compare these samples with each other. So I guess a challenge would be to see if you can do this using ggplot. Line 54. So I mentioned that this is an S4 object, which is a really efficient way of including disparate types of information, but it can be a little tricky to use. So this line 54 shows you how to extract, for example, information on how the data were processed. So you use this @ symbol to extract the relevant information. And if we run this, we see that for each of the samples, the data were analyzed with Partech Genomics Suite 6.5 using the RMA algorithm. So the RMA algorithm, so that stands for Robust Multi-Array, and it's a pre-processing algorithm that tells us that these data have been background corrected and quantized. So that's quite important to know because if you aren't dealing with pre-processed data, you would have to do the pre-processing yourself. Or maybe you don't like the way the pre-processing algorithm they've used, so you can change that if you wanted. So great. So that's all we have. That's what we need to do to get the data into a form where we can actually do the differential expression analysis. So to do that, so that starts on line 61. We first need to set group names. So that's what this paste function does. And if we type it out, so that's SML. It's just two groups, G0 and G1, and within each group there are 3 samples. Now, to do this Lemma analysis, what we first need to do is make a design matrix, and I'll briefly describe what that is in a minute. Now, for the design matrix to work, each of the columns of whatever goes into the design matrix has to be a factor. So we need to convert this SML into a factor, so we use the as.factor function and save it as another object called FL. And if we run FL, we see that it's like SML, but now it has 2 levels, G0 and G1. So in GO2R, what they do is they assign FL to a description column within GSet. We'll go ahead and do the same thing. And you use this description on line 65 to make your design matrix. So I'll go ahead and run that, and then we'll take a look at the design matrix itself and discuss what it's used for. So if we click on design, we see that there are 6 rows, and the rows correspond to the samples you're using. So the number of rows has to be equal to the number of samples you're analyzing. And you see that there are 2 groups, description G0 and description G1. Now, these 1s and 0s are dummy variables. And what it's telling Lemma is wherever you see a 1 in the first column, those samples belong to group G0. And wherever you see a 1 in the second column, those samples belong to G1. Now, there are other ways of making design matrices, and I'll show you an alternative way, which I actually prefer, and I'll explain why I prefer that. So what Lemma does is it for all the, in this case, all the probes, it's basically just fitting a simple linear regression model to perform this differential expression analysis. Now, in this case, we're comparing just 2 groups, group 0 with group 1. So our linear regression function will be beta 0, which is the intercept coefficient, plus beta 1. And in this case, beta 0 refers to the mean gene expression in group 0, and beta 1 refers to the differences in the mean expression between the 2 groups. So Lemma, what it does is it just does a simple t-test comparing beta 0 plus beta 1, which is the mean expression in group 1, versus beta 0, which is the mean expression in group 0. It's probably best to actually see this all written out. And so I recommend looking at the documentation for the Lemma package itself. It explains it really nicely. So if we go back to the code, line 66, we give the, so right now, the design matrix has these are the column names. So we can change the column names here with line 66. Actually, not sure how it changed, so that's not super important. If we go to line 67, we fit the first linear model, and that takes the G set, which contains our expression values and the design matrix. We save it as an object called fit. And then what we want to do is we want to create another matrix called the contrast matrix. What the contrast matrix does is it specifies what comparisons we want to make. Now, in this case, it's obvious we're only making one comparison, G1 versus G0, group 1 versus group 0. But the contrast matrix is especially useful when we're including additional variables or covariates. And we're interested in making, we're interested in certain comparisons within this larger model. So we'll go ahead and make the contrast matrix. And we can take a look at it. That's what it looks like. And so then we do a second fit. We do use this contrast function, and that takes two values. One is the fit, which is the model we've already fit, and then we use the contrast matrix, which provides the comparison we're interested in. And in line 70, what we do is we use an empirical base approach. So what this does is, so as you know, when you do a t-test, you first need to create a t-value. And to do that, your denominator has a standard error of your gene expression. So what empirical base does is the approach is it takes into account the distribution of variances of all the genes of interest, and it works out a weighted mean of the mean of this distribution of variances and the variance of your gene of interest. What all of this does is it ends up shrinking the variance of genes that have high variances. And by shrinking the variance, you shrink your standard error. And by shrinking your standard error, you get a higher t-value. So you increase the power of your experiment by using this empirical base approach. And what it also does is it also increases the number of degrees of freedom you have. So that further increases the power of your experiment. Again, this is also nicely explained in the documentation for the Lemma package. So we'll go ahead and run this ebase function. And you'll see all it takes is this fit2 object, which we made in the previous line. And this another argument is just 0.01, and that refers to the assumed proportion of genes which you expect to be differentially expressed. I actually don't think this has an effect on the outcome, but 0.01 is the default. You expect 1% to be actually differentially expressed. We go ahead and run that. And then finally, we use a top table function to extract the variables of interest. So that takes in, so we'll use the fit2 object, and since we're doing thousands of pairwise comparisons, we have to include some kind of multiple comparison correction. So that's what this adjust argument does. And so we're using, FDR is using the Benjamin-Hochberg correction, which is a more conservative post-hoc correction. We can also do the Bonferroni correction, which is very, very conservative. And so in GO2R, it sorts by this B. So this is it's a log odds value that gives you a measure of how likely this specific outcome is. And number equals 250, it just defaults to the top 250 genes in descending order of significance. And we can adjust this number to make a larger data set. And we run that, and we get, we save it as an object called TT. And you see that the first few columns have a lot of information on the genes themselves. So you see the top gene is the gene called ABCB1A. And if we scroll all the way to the right, we can see that there's a log fold change. So there's log 2 fold changes, t values, p values, adjusted p values, and so on. So this is, as I mentioned, this is in descending order of, or rather ascending order of adjusted p value. And just to confirm that we are getting sensible data, we can actually look at, so as I mentioned at the start, we're comparing wild type with this TSE1 knockout. So we'd expect TSE1 to show reduced expression in group 1 versus group 2, and it does, which is great. And TSE1 is a tumor suppressor. So if you look at, for example, Ki67, which is a cell proliferation marker, if we look at it, it goes up. So these are just checks to see that if the analysis you're doing makes sense. So the second set of lines, so from 74 to 81, is the same as what I did previously, but it's just a different way of making the design matrix. So I call this DesignAlt. So in the first case, so if we look at line 65, we use the model.matrix function to make the design matrix, and we tilde description plus 0. In line 74, we do the same thing, except we dropped it plus 0. So if we run that and take a look at DesignAlt, it's just taking a second. So if we take a look at that and we compare it to design, so this is what it looked like. So it had a 1 for all the samples that belong to group 0 in column 1, and it had a 1 in column 2 for all the samples that belonged in group 1. If we look at DesignAlt, it's a slightly different look. It has the values for the intercept and the values for the beta 1 coefficient. And I like this because this reminds me of the linear regression function, beta 0 plus beta 1. Now, all the linear regression equations for all the samples have the intercept. That's common to everything. So there's a 1 for all the beta zeros. But for beta 1, which, as I mentioned, is the difference in the mean expression between the two groups, it has a 0 for group 1 because that's what you're comparing it to, and it has a 1 in group 2. So group 1 is just beta 0, and group 2 is beta 1 plus, beta 0 plus beta 1. So the t-test you're doing is comparing beta 0 with beta 0 plus beta 1. I just find it easier to keep track of what I'm doing using this design matrix. And the rest of the code is almost identical. So another difference is how you do the establish the contrast matrix. So in this case, in the previous case, it was but it's design. It's easy. You're just comparing column 1 with column 2. In the second case, you're comparing the 1s in column 2 with the 0s in column 2. So to do that, you just use a make contrast function, and you just give it the name of the second column, which is description G1, and the levels design_alt to run that. And it gives you a warning message. It tells you that it's renaming intercept to intercept, which is fine. We run everything else, and we get TT_alt. Now the TT_alt and TT should be identical. They aren't quite identical because of, I think, like this thing called floating point error. But if we compare the IDs, which are the gene IDs between TT and TT_alt, they should be the same. We use an identical function, and it says that it is true. So the output is the same, whether you use this design matrix or this design matrix. And I also have another thing where I say, so previously we extracted an object called X from GSet using the EXPRS function. You can also use that object to do your differential expression analysis. The only thing with that is you don't get all of these additional columns. But I actually find it easier to work with this EX object. And so I won't go through that, but if you want to use that, you can just use the same code that we've used previously. And so if you're happy with that, you can just save your dataset using this sub. So you subset the TT dataset to choose the columns of interest. So again, this is a base-hour way of doing what we're used to. So this is a base-hour equivalent using the select function in the tidyverse, which is what we're used to doing. I just saved this, use this as is, because this is what is in Geo2R. But as an exercise, you can go through all of this code and convert it to the tidyverse format. And then you use the write.table function to make the table in your working directory. That's it. I hope you enjoyed this. If anything is unclear, especially with the Lemma processing, I again strongly encourage you to look through the documentation of the Lemma package and/or email me. Thanks for watching.
Thank you, Akshay. That was certainly deserving of an applause. Quite impressive for an experimental biologist taking us through a deep dive into some heavy statistics. I'll just sort of highlight two important points. The first is, if any of this information in the statistics is overwhelming or quite a lot, what I would encourage you to do is just to keep at it. So when we started our own data science journey, everything was overwhelming. And every time that you expose yourself to it and every time that you learn something, you will get a little bit more out of each of the lessons. And so you can go ahead and watch this lesson again or tune into other lessons, but I would encourage you just to keep at it. So we have a few minutes if you guys would like to ask any questions to me or Akshay on the message board. But otherwise, thank you for coming along with us, and we hope to see you again next month.